Hey, what's going on? He's another one that asked me good questions that makes me think. Okay, so I'm going to stand by that. Yeah, but less than 130 years. I'm just going to say about 100 years. It has to be before Seth was born. Okay. So that's your question on the bottleneck. What do I mean by a one generation bottleneck, you mean?
Right.
Okay, so good question. According to the biblical model, there would be essentially 3 bottlenecks. You got one at creation with Adam and Eve. You got one after the flood with 8, Noah, his wife, Noah's 3 sons, and their wives, 3, 6, 8. The Bible says by the 3 wives of Noah's sons was the earth repopulated and inhabited. So they would be technically, if you're doing the models, you're gonna have to start from 6. So there's your second bottleneck. And your third bottleneck, in a way, I guess, at the Tower of Babel, if you had everybody, let's say at that time, 10,000, 15,000, somewhere in the thousands of people all living in one place at the same time, that would have dispersed. So in a sense, that would be a bottleneck too. So what I mean by one generation bottleneck is, okay, you got Adam and Eve, and they start having kids right away. It's not an extended bottleneck. I mean, you've got, you start with 2, then 4, and you keep going up. It's followed by rapid and exponential growth. And same with the bottleneck after the flood. It's followed by rapid and exponential growth, as well as the Tower of Babel. So when it comes to like the evolutionary out of Africa bottleneck, you've got, I get conflicting answers because, you know what, which is understandable, because how can you know exactly how much? But I've heard 2,000, 10,000 roughly was the bottleneck. But it wasn't like this 2 to 10,000 in the very next generation or the next even 100 generations experience rapid and exponential growth, where you've got 100,000, now you got 500,000. They stayed between 2 and 10,000 for hundreds, maybe thousands of generations.
Yeah, that's a part that I'd never heard of, and I don't know. For a while, I thought maybe you thought they didn't like sex after they came out of Africa. I don't know, but
no, I'm sure they enjoyed it. Well, okay, let me ask you then, because that's another question I find I don't really get a straight answer from, and I've searched heavily. How many generations was the bottleneck? You got a near extinction event that sends the human population at the time to between 2 and 10,000, let's say. How long did that populate? Because remember, it has to be, especially from the papers that I've read, and Francis Collins, who talks a lot about it, and Dr. Vanima in his book Adam and the Genome. How long was it? Because it has to be prolonged, because they're invoking the population bottleneck to explain low human genetic diversity. They have to reduce the genetic diversity, and that would happen based on generations and generations of inbreeding and mutation accumulation and things like that. So what do you think? How long was it?
I don't think there was. I think the bottleneck was like for each tribe that came across, like they probably came across in groups of from 20 to 100. They had a great deal of you wouldn't call that incest, but there, that was a viable breeding population. But I'm sure that they had, when they crossed over into an area where there was game, where there was basically they could populate very quickly, and I think they each spread out in their own tribes, in their own groups, and they could have. I don't know a lot about paleontology, but they could have interbred with Neanderthals at that time.
Yeah, it looks like they did.
And anyway, I don't see that the bottleneck was long. It wasn't a long bottleneck. It was just a sort of a one. The bottleneck.
Would you say it was more? Well, it was a lot more than one generation, like our bottleneck. You would admit that at least.
No, not at all. I think there were a lot of little bottlenecks that came out, in spurts, as they came across, and they dispersed probably pretty widely. And when they dispersed, they had resources.
Okay.
I don't know.
Okay. Well, wait a minute. So you've got Africa, you've got Homo erectus, like all the Australopithecines, your early Homo variants, your Homo erectus, for example. If you want to consider Homo habilis as well, Homo ergaster, whatever, it was Homo ergaster, just a variant of Homo erectus. These were all in Africa, even according to the evolutionary model. So you've got Homo erectus evolving for millions of years, accumulating more and more mutations and therefore more genetic diversity. And Homo erectus was evolving from some pre-Homo groups, and before that, some Australopithecine groups. So that's millions and millions of years of mutation accumulation and genetic diversity increasing, for example. So we know today humans have very low genetic diversity. So that population bottleneck, that near extinction event, you'd agree it occurred in Africa. So are you saying that a lot of these groups then would have formed tribes within Africa? They would have moved to different parts of Africa, is what you're saying? Not like out of Africa.
Right. There were a lot of them that spread in Africa and basically stayed there, and they have less genetic diversity, or more genetic diversity than the groups that came across into Europe. And I don't know enough about all of that to develop a really good model of it. But what I've been able to glean is that, it happened over a period of maybe 1,000 years, that there were all these little groups that came out, and each little group had its own genetic cocktail.
Right.
And they spread, and then they started, and of course, some of them started mixing, and we would have, it would be an incredibly difficult model, but I don't see.
Here's a good question. The bottleneck today that the cheetahs are experiencing in the world today, you'd agree that this has been a prolonged bottleneck?
No, I don't know why you say that. I don't
because
know
this is what the evolutionists will even admit. This is what
I've never read about.
Mike, think about it. No, but think about it. This is the only way to reduce homogeneity. The reason they're invoking this, like, for example, did you follow what I was saying? Does that make sense that if you've got Homo erectus evolving for millions of years in Africa, all of these generations, you're going to accumulate a lot of genetic diversity. You're going to accumulate a lot of mutations. So why do humans today have low genetic diversity? Well, because of this this near extinction event, it had to occur. This is what the evolution is. I'm not just making this up. It had to have been prolonged. It had to have been many, many generations in order to reduce the homogeneity. That's the only way.
Okay, think about it this way. You've got an aquarium, and in the aquarium you've got 10,000 goldfish, and they all have just a lot of diversity. There's a lot of different kinds of spotted goldfish that you have there. And then they start to die out, and they start interbreeding a little bit. And then you have maybe 20 or 30 different spot types. And you dip in there with a dipper, and you pick out 6 of them, and you set them on your own. And you're going to have you're going to have a very limited set of genes. Okay. And then you put them into their own tank, and then they take off and start multiplying because now they have resources. That's how I view the out of Africa scenario. I don't see any rationale for your saying that it's a prolonged bottleneck. And I don't. I'd have to look at the temperature, the effective extreme.
Well, Mike, even the cheetahs today, the 10,000, they're down to like 6,000 or 7,000. It's not just 6,000 or 7,000 that are just all chilling in the same place, like some one world government, and you got your one world cheetah leader. So obviously during the out of Africa population bottleneck, sure, you're 2 to 10,000. Many could have been spread out into different smaller tribes. That's just going to make the inbreeding problem even worse. Oh, so go ahead.
Yeah, there were like 20 to 100 people in a tribe, and they
even worse. Even worse.
Well, I don't know if they'd survive at any smaller than 20, but I'm pretty
But
sure.
remember that I got to point out this though, and you probably understand this now studying our model. We see, the thing is, you can't argue again. I'm not saying you do, but a lot of people, they put forth arguments, but the arguments are not carefully thought out, because they're almost arguments against a non-position, because the biblical based model would say that Adam and Eve were created with no mutations. Now, inbreeding becomes a problem because it's the exposing of, as I always say, the hidden reservoir of genetic mistakes. But if there are no genetic mistakes to come to the forefront to result in genetic damage, for example, well, you're not going to have a genetically damaging related bottleneck. But with the hypothetical out of Africa population bottleneck, as I said before, you guys explain all DNA differences, all genetic diversity as a result of mutations over time. That means you have millions and millions of years of mutations accumulating, most of them neutral, as you say, right? That's kind of like, I've kind of adopted your explanation where those neutral mutations are absorbed by the junk areas, and they're kind of used as a hidden reservoir for evolutionary advancement in the future, right? They're kind of called upon when necessary. But you're accumulating all these mutations over time, and then all of a sudden, boom, this population of humans at the time, Homo sapiens, was reduced to just 2 to 10,000. Now you have all those mutations that have been accumulating for millions of years. They're coming to the forefront. Boom, boom, boom. They're popping up. They're leading to genetic damage, like with the cheetahs today.
Okay, you're using an argument that I totally do not agree with, this genetic entropy argument, and we're basically veering off the point with that, because I mean, I could crush that crap. So we don't want to use that. That has nothing to do with it. The thing is, when you dip into a population, and if you take a small group and you move them into a new geographic area, that is called a bottleneck, and that's happening right outside in the river valley in my backyard. There are little groups of insects that are being cut off by maybe a sandbar or something like that, and they end up in a small population, and that's called a bottleneck. And it's not a problem as long as, for cheetahs, for instance, I read that it went down to 7 individuals. I don't know how they know that, but there's been, but I mean, 7 individuals could possibly create a viable breeding population.
Well, it would be inference, I'd imagine, based on the known genetics currently. I'm not saying it's a bad inference. I'm just saying they got to look at, like, that's what they do today. They look at the levels of human genetic diversity, and they know that it's low. We're all incredibly similar. Me and you, we're 99.99999% similar. So
I'm studying bioinformatics, and I'm finding just hundreds of variations. Well, there are I forget how many million or 100 millions of genetic variations in individuals today because of our population size.
Well, me and you have within our own genetics, we have about 3 million places, more or less. I don't want someone to come on and say, we got 2.8 million or something. Okay, more or less 3 million places where the copy we got from our mother, or the copy you got from your mother, differs from the copy we got from our father. So, but in the world, you've only got about 10 million common variants. So if we individually carry 3 million, take those 10 million common variants, throw them into Adam, and boom, all your world's diversity, all your models, all your population genetics flow perfectly from, because you find those 10 million variants, you find them in China, you find them in Africa, like we're talking about, Europe, you find them in the entire world, which means actually it fits nice with our model, because those variants would have had to be, if they're common variants, this only makes sense, they had to be in the population before we spread out. That means, according to our model, boom, those variants were on the ark.
No.
Ultimately, they were in Adam. Well, do you disagree that there's about 10 million common variants in the world?
No, I mean, as soon as the population was sampled, okay, once you take that sample of population and you have a certain genetic profile, right? You have a certain number of alleles. And right after that point, every generation adds new variants. And we've been adding variants ever for 80,000 years.
Well, the mutations would create new variants, but you get new chromosomal and allelic combinations just through recombination.
Yeah, but there's a problem. I mean, you can only, okay, when you have two individuals, you have, at most, you have 4 alleles. And for something like insulin, it is not a good idea to have variation. Of course, there is variation that doesn't matter.
Well, real quick, I seen you define, I think it was you or Immutable Destiny. You defined allele correctly. It was in one of your hangouts. What's your definition of an allele?
And an allele is basically a locus or loci.
It's a gene variant, but like.
Well, it isn't necessarily a gene. No, they call alleles. They do it a little finer. It can be even a part of the gene. But let's just say it's a gene. So you have one gene for insulin,
right?
And you have up to 4 genes for insulin in a mating couple, and then there's a recombination event, and you get a mix of those. The possibility of those 4 genes, but for that allele, you're going to pick one of them, or 2 of them, only 2 of them. Okay.
Okay, but you said, so it looks like you're defining an allele at a genetic loci. Well, what's a loci?
That is a particular a spot for a gene. There can only be one spot for one gene. You can have
it's a location on a gene. So how many locations? How many genes in our genetics, and how many possible locations in our genetics?
Well, okay. So you got one gene for insulin, right?
Right.
Okay. It can only have, in a mating couple, it can only have 4 alleles for that particular gene. Now that doesn't mean that the gene could be copied somewhere, and now we've got 2 copies of the gene. And that isn't the case for all genes. For some genes, there are only those 4. And out of those 4, 2 are going to be picked according to some probability, and that's Mendelian genetics. So what you're relying on is sort of an interaction between a number of different genes in a different gene cocktail. And I would have to work that out very carefully for phenotypic traits. For instance, like Matt going on about skin color, I think he's just all wet on that. There's about 7 variants, I think, that go into what color your skin.
Yeah, but we'll get into the skin color, because so if you're it goes back. So the variants you're talking about in insulin, okay, so that's one example. But if you're defining allele in terms of genetic loci, a spot on a gene, a specific location, you've got nearly unlimited locations. So if you're going back, as you said, Mendel, if you're going back to that idea of big A, big B, little b, now, I don't know why anybody would ever assume that Adam and Eve, even if you don't believe it, I mean, if you're putting forth a model, why would you assume that Adam and Eve were just created with no genetic diversity? They were just created genetically homogeneous. God said, be fruitful and multiply. I mean, that would make no sense. Well, what makes sense is that God would have created DNA in their DNA variety in their genetics. And real quick. So that means if God created Adam and Eve with, as we're saying, if he created them heterozygous, and we're defining allele in terms of genetic loci, which we know there's a lot of locations in our genetics, that means you've got capital A, lowercase a, capital B, lowercase b. But guess what? You're not just doing that for one location. You're doing that for the millions of DNA locations, DNA spots that actually exist. That means starting with an Adam and Eve with that created heterozygosity, that means they do have the potential to produce all sorts of variety in even a single generation. Then you can build your model. You can look at the HapMap data, for example, look at how many common variants there are around the world. Look how the allele distributions are, for example. I don't know if you've read any of Rob Carter's work where he's examined the HapMap data.
Try not to.
But it does, you should though, it's interesting. I've got a paper right here. You'd actually enjoy this. Lots of charts, lots of, he starts, I think that's beyond the scope of this discussion, but Like, does that make sense to you? Because you even admitted that you're defining allele in terms of genetic loci. So
Right.
you have to apply that now to the millions of DNA positions, loci that actually exist in the genome. All of that's being reshuffled even in a single generation. Go ahead.
We're talking about one trait or one protein, like
Right.
insulin. There are only 4. There's only one loci
Of that one trait.
of that one trait. Now you're talking about a combination of traits, and you're kind of hoping that there's a way for this combination of traits to create all this diversity. But I don't think that works. I think if you work it out for most, like even raising collies, like the long nose of a dog, I believe was from a mutation, not a different combination of, not epigenetics and not a different combination of genes.
Well, it's not really that difficult, because what I like about you is you do have the accurate science, so you're easy to talk to. Most people, when they use that argument, they're defining allele as a gene unit, and that's just not the case. You can even see this in the secular literature. So when I seen you in Immutable Destiny define it accurately, you defined it in terms of single genomic position, I thought to myself, okay, this is great. Now they can understand. Look, let me just break it down. You've got a single gene, okay? Typically they span, it's not like this every time, but say thousands of nucleotides, okay? You got these single nucleotide variants now that would be distributed also throughout that gene. So let's say you have even 80 or 90 of those SNVs spread out within that gene. And now you're looking at the genomic position definition of alleles, like you are doing accurately, and I'm thankful for that. Every single one of those 80 or 90 or even give it 100 single nucleotide variants could have existed in a heterozygous state in the kinds. If you want to go to the ark, that would have existed in the pairs brought on board the ark, would have existed in Adam and Eve at creation, for example. I mean, even if you don't agree with it, that is a tremendous amount of allelic diversity. I'm
not sure if you're quite getting this or not. But for one gene, okay,
yeah.
And if we consider one loci as one set of alleles, there can only be 4. And brought on the ark, there can only be 4 for every animal
of that trait.
Adam and Eve. For that particular gene, now a trait can be made of multiple genes, and that would save your argument a little bit if you could figure that out. But I don't think you're quite thinking this through, and that's why we always have a problem with your created heterozygosity, is that if you have 80 SNVs in one of those genes and 80 in another and 80, times 4.
Okay, well,
you still are only going to have 4 of that particular flavor of gene.
Now remember, the evolutionist, is assuming that all these traits, all this DNA variety came about over millions of years of mutation accumulation. Okay. Now, when you have all those mutations accumulated, yeah, you can have a generation of recombination or gene conversion, and that's going to produce new variety. But that variety, according to the evolutionists, is there because of millions of years of mutation accumulation. But according to our model, if that DNA variety was front loaded into Adam and Eve, that means just basic Mendelian genetics. You've got, like I said, big A, big B, little b. Those letters constitute different features and different traits. So yeah, you're pointing to one trait. How many millions of traits actually exist in the genome? Why do a family, why does a husband and a wife have a child who has red hair? But yet you see red hair was a trait that existed in that lineage 10 generations prior. That's because a lot of these traits are in recessive hidden form. So that means Adam and Eve would have had a multitude of traits at creation in hidden recessive form, just like we see. Even you look to pre-existing heterozygosity. We only accumulate 100 new mutations per person per generation. Everything else is pre-existing. Everything else is inherited. Correct?
If that red hair is a result of one gene, then there can only be 4 variants. and those four variants can combine. You could have an A, and you can have, or as you like to say, an uppercase A and a lowercase a. You can have two versions of that gene.
Well, because if you had two uppercase A's, well, you're not going to have much variety there. That's homogeneous. So of course you want a lowercase a and an uppercase A, or a lowercase b and an uppercase B.
And they're going to be slightly different. Like for insulin, for instance, or for one particular gene, you can have 50% of those. You can have 50% of it can vary SNVs on 50% of the nucleotides and still have exactly the same function, exactly the same trait. Okay, so it's not buying you a lot of diversity. The reason that we believe in it, and you mentioned that we use mutations as evidence for our evolution and for our change. The reason we can do it is because we deal with populations, and populations are going to be much bigger than two individuals. They're going to be usually around 1,000 to 7 billion or whatever. So I mean,
you would need large of populations, because in order to explain the diversity, in order to explain the traits, in order to explain the genetics we see in humans today, you would need a starting population of thousands. But that's, like I said, that's also, and I think this is where your the hindrance is, and it's understandable, because that's your position, that's kind of what you hold to, but that's assuming that mutations are the source for all DNA variety. I'm telling you, if you look at these DNA differences, millions and millions of DNA differences that exist in our human genetics, if those were built in and front loaded, and we can figure this out, because a lot of times it was Emo who asked me the question. He said, well, how can we tell which one's created, which one's not? Okay, we just have to look at allele frequencies. Those that are common, those that are not disease causing, those that are functional, those are most likely the created ones. The ones that are rare, those would be the ones that are mutations, because those are the ones that are not common. Those are the ones oftentimes that are geographically specific. And I'll send you a bunch of these. There's one really, really good paper, extensive, that goes over all this modeling based on allele frequencies, looks at the rare ones, looks at the common ones, and kind of shows what Adam and Eve's original genomes would have looked like. But you can't assume that all those DNA differences are the result of mutation, because then what you were saying with the Adam and Eve, with the skin color, with the races, well, then it makes sense, because if you had a mix of the capital and lowercase letters, not just capital A, capital B, capital B, like a mix of them, that means Adam and Eve could have been a middle brown skin tone, and now they do actually have the potential to produce every single shade of skin that exists on the planet. That's if you assume the front-loaded DNA variety, though. So if you assume it right now, Mike. You have to argue it you got to put yourself in our position. Okay. Assume the worldview. Boom. Assume it. Now argue against it.
Well, that's why every time you mention heterozygosity, your opponent wrinkles his brows and raises his eyebrows. You get the raise, because it would be very difficult to model that. And it seems like you're missing something here. I think you are not understanding something about alleles and how they work. And CRISPR tried to point this out to you, that there are only 4 of those alleles available to Adam and Eve. And what you seem to be saying is that these genes can be in more than one place than they are, and they can't.
Okay, well, how many genes do we have?
I don't know what the number.
Okay. Well, I'm not trying to put you on the spot. I'm just okay.
I think with 100,000 protein products.
Right. Well, we've got, only 2% of our actual gene, of our genome codes for proteins, for example, and most of it is non-protein coding. But the point is, if you're, as you are, defining allele in terms of DNA position on a gene, okay, a DNA loci, well, how many locations? Just give me a number. How 4. Many DNA locations exist in our genome?
For one set of alleles, only 4 at most in a mating couple. You can't. Well, unless the gene is doing.
Yeah, but now you're contradicting yourself. So now you're defining allele in terms of gene unit. Well, what is it? Are you defining allele in terms of gene unit or genomic position?
The word allele means a difference at a loci,
right?
At one particular locus.
Yeah.
You have this.
So how many differences? How many different? You could have millions and millions of differences in chromosomal pairs in the genome. There's a lot of DNA variants in the genome.
All of the genes, but that isn't how you define an allele. An allele cannot mix any more than at that one locus.
Right.
But 4 types at most in a mating pair.
You have to take that 4 possible variants at that loci. Now you have to apply that to all the millions of DNA positions that exist in the genome. I don't know why the 4 is more.
How does that work for one gene? I mean, for one trait. Okay, one trait is set up, by some cocktail of proteins, and I'm guessing, okay, we could say 30 different proteins go into this one trait. Skin color, I think it's somewhere between 4 and 8 that go into one trait. Okay, so for skin color, okay, granting you 8, you could have 32 variations, and and it is possible that would work. But I think you keep sliding between this bad definition of what an allele is. An allele is
you defined it as that first. That's why I clarify. You defined an allele in terms of genomic position. We know there's not just 1 or 2 or 4 genomic positions in our genetics.
Allele only refers to that one position. When you're talking about allele variation, you are talking about that one position. And you can only, in a mating pair, you can only have 4. Adam and Eve could only possibly have 4 for that one gene. Okay. Now, what you're saying, okay, let me try to steelman your position. In all of this extra DNA, this 98% that is non-coding, there are also all sorts of little widgets that basically all combine together to form this multitude of traits. And you're saying that basically all variation is a result of what was originally in Adam and Eve. You can do that, but don't talk about alleles, because you end up with that problem that we're talking about one locus, and you can only have 4 variants in Adam and Eve. Okay, no more than that. You have to correct that in your thinking. You have to get down with some paper and draw this out, because you're not saved by saying that, like for insulin, for instance, you're not saved by saying that there are 5,000 different places where insulin can vary. And that would amount to with insulin, I'm guessing that we have about 120 variations that would be called SNPs, single nucleotide polymorphisms.
Are you saying in the entire population, like in the entire world population?
You're not going to get that. You're not going to get that out of created heterozygosity.
Of course,
you're only going to get 4 of them.
So. But that's why I said, if you look at how many family trees, okay, have been produced since. Well, let's start from the ark. How many family trees have been produced? How many mutations have been accumulated since Babel and that dispersion? So yeah, a lot of those variants are the result of mutations. Yes. That's why you have to look at allele frequencies to decide and figure out which ones are created, which ones are the result of mutations. And that's why you can look at the ones that are more common versus the ones that are rare. But real quick, because what you were saying, if you take the genomic position definition of alleles, is what I'm taking here, not the gene unit. We have to define it in terms of a single genomic position, but it has to be independent of its relationship to a gene. Okay, so if you expand this single example, this single gene example, we expand it throughout the entire genome. That is going to reveal an extraordinary potential for allelic diversity, even if we start at the ark, even just on the ark. Because in 2 diploid individuals, as you said, what'd you say? 4 genome copies exist, right?
I just created a red flag, you just gave me a red flag. No, you can't do that. You can't do what you're doing. You can do that with a phenotype, but you cannot do that with alleles and with diversity. And I think you guys need to work on your model a little more seriously, because that's why we always wrinkle our brow and we don't know what the hell to say to you about created heterozygosity, because it just doesn't make any sense to us.
No, you see, the thing is, you can even read secular literature where they're arguing over the single genomic position definition or the gene unit definition. And the fact is, the best way to define allele is in terms of genomic position. So if you have only 4 DNA base pairs that exist, now you have virtually every possible genomic position allele, which is far more than the 4 to what did you say, 48 gene unit alleles. Those are what have been present at, I don't know, maybe you haven't thought through the genomic position definition. I mean, you're the one that defined it as genomic position. Are you saying there's only
4
at a position?
2 in one individual, right?
How many positions in our genome is the question? A lot.
For that one gene?
No. Okay. So for the insulin gene, okay.
Yeah.
You don't need, we're trying to say all of these traits. I mean, you're looking at traits for hair color, you're looking at traits for eyes, you're looking at traits for immune system, you're looking at, I mean, there's a lot of traits that exist in our genetics. That's what Mendel looked at with the, what was it, the pea plants? You know, yeah. He looked at dominant and recessive traits, and a lot of the traits are in recessive form. The ones that are in recessive form, you would say, correct me if I'm wrong, those are still the result of mutations over time. They're just in recessive form. Adam and Eve at creation would have had millions of recessive traits already front loaded into their genetics, just waiting to be revealed.
Yeah, but all different traits. I mean, they wouldn't have had combinations loaded for that one trait.
Right. So your problem is the insulin trait. There's about 120, for example, that exist in the human population. The SNVs, right? Single nucleotide variants.
Yeah.
Right. Okay. Yeah. And I'm saying that the ones that there are that many of course, we're going to expect mutations over time. I mean, now we're probably just going into a circle, but I do want you to read this. I have it in front of me, very, very technical. I mean, have you looked at the HapMap data at all?
What HapMap data? Which one?
Let me see, just real quick, because he's running.
Which database?
Models here.
Can you send me some links to that stuff? You know, maybe we'll do a debate on this at some point after. But please provide me with some of your links to this Carter's work. And
even the diet. I mean, I kind of like this respectful. I mean, we can probably go on forever on this, but you know what? That I'm glad we got into the. I know you're saying that I need to rethink some things, but I think you just need to rethink instead of just saying, okay, yeah, because you're the one who said it at first, you're defining an allele in terms of genomic position. You just need to now rethink. You need to approach the data now with that assumption and starting point. But I think on that topic, we've probably ran it dry. But here's one question that just came to mind because of what we were talking about. And not to put you on the spot here, I'm actually genuinely curious, because these are certain questions that I've had for myself that I'm just like, man, how could I, because I want to assume the model, okay? And that's why I'll oftentimes point out like what I think is some of the best lines of evidence for universal common descent being nested hierarchies in highly conserved mitochondrial DNA proteins like cytochrome C. Why do all these genes form nested hierarchies? You know, I'm assuming the model, and I'm going to try and argue against it with the model. You know, I don't want to argue against a straw man. So when it comes to, because you're talking about all these proteins, if you look at alternative splicing, which one single protein coding region of our DNA, and I find this fascinating, I've talked about this for years, can literally give rise to hundreds or even thousands of different proteins. I mean, this is far different than the one gene, one protein, one function from years ago in the 40s. How does evolutionary processes evolve such a thing? Like,
I
what do
think
you
100 is the max, and I think that's pretty rare. On average, I think it's 6 or 7, or is it 4? I think it's 4.6 proteins per gene because of splicing, and that's something that only appears in eukaryotes. So
Okay. Well, so eukaryotes versus prokaryotes, yeah. So, but how does evolution give? Okay, the fact is, it can give rise to hundreds or thousands. Maybe the thousands isn't the rule, but even 4 or 5 or 10. I'd have to look into that to see for sure. But regardless of the number, how does that? What type of evolutionary process evolves such a thing as alternative splicing?
that's a whole nother subject.
Right. No, and because this isn't, we're just dialoguing. Like, these are questions I have that I'm like, okay, because there's been so many. I'm not the one making the predictions, but you can read. I'm sure you have it because you said you don't like to. I have it here. I should just put. I've got a ton of papers here. I've got the Ventura paper here. I thought we were going to talk about, but we can talk about that later.
2012.
The Ventura.
Yeah, 2012. That one.
March 2012. Yeah, but real quick, have you read the one that I sent you by Jensen? And I know you don't. It'd probably give you a headache, but just do it for me. Recent functionally diverse origin of mitochondrial genes from more than 2,700 metazoan species. Have you read that paper yet, where he goes over the predictions on DNA function, even the neutral variation, the cytochrome C? Have you looked that over?
Yes, I have. Yeah.
You have?
Yeah, but I can't remember too much about it. That was from last week or something that we did that. Not entirely sure.
It's one of my favorite papers because it's what has set me on, I guess, a lot of rabbit trails going into these things on DNA function. And it's just like even the redundancy, for example, that requires forward thinking, because it's natural selection. And I think I mentioned this to RJ and Erica. Natural selection works on the short term, not the long term, but yet we have all these redundant mechanisms. You know what I mean? Like, don't you think to yourself, wow, how did evolution build a spare tire? You know, every time I see a spare tire, I'm working on a car or something, I'm like, evolutionists must look at this spare tire and think, look at this junk, look at this evolutionary leftover. No, that requires forward thinking. That's why that spare tire and that jack was put into the car in the first place. You get a flat tire on the road, boom, thank God you've got that extra spare tire. So what type of evolutionary processes evolve something like a spare tire?
Okay, if you want to use cars, my first car was a '62 Chevy Super Sport with a high performance 327.
Nice. You must have been picking up the ladies.
We tried.
We tried.
They kept getting away. But anyway, if you can imagine an entire junkyard in your trunk, and then every time something went wrong with your car, you could go open your trunk, look in the junkyard, and find a little piece, hobble something together. Like when you talk about junk DNA, I don't think anyone ever thought it was junk. We thought it was a junkyard. And if you come from the 50s and the 60s of the last century, as I did, junkyards are places where you go to build a car. They're basically, or where you go to modify your car.
I look at the junkyard like the fossil record for the evolutionists. They're going to go and say, what is this? And what is this? These must be leftovers from all our past common ancestors. I could go to the junkyard and probably build, I could line things up and build nested hierarchies and stuff. And I want to hear more about the car, what you're saying, but seriously, I am curious, what type of evolutionary process evolves that which requires forward thinking, like redundancy in our genetics? I'm just using a spare tire as an example, because that would be like a redundant element in a car, but we know it's not. It's there for a purpose. Like, what type of evolutionary process evolves such a thing?
Got it. car analogies really piss me off. But
okay, forget I use the
no, I can go further into this.
Forget I use the car analogy.
No, you got me going.
Redundant mechanisms in general, you know.
Let's do cars.
Do cars. I like cars. cars.
Suppose I have a car made out of Lego blocks, basically. That there are 1,000 different kinds of blocks, 1,000 different Lego blocks, and in my trunk, I have this system that keeps, throwing them together and turning them around because I'm going over a bumpy road, and it keeps knocking them together and forming different combinations. And then I'm driving my car, and all of a sudden I cross over into the twilight zone, and I'm in an area where all of a sudden, oh my God, my car is no longer on the road. It's not working. I have to go fix this. I go back into my trunk, and I get some of these Lego blocks, and I make some sort of a device that allows me to run my car over this new kind of road, and all of a sudden I've got a speciation event. That's how it works. Okay, it's a bunch of Lego blocks that all pop together. But I think we have so much work to do, you and I, and I don't think we can do too much more tonight.
No, and I see my wife take the kids outside because they started crying.
Right.
2 hours. I didn't realize. I'm like, yeah, no, don't worry. The kids just need an hour nap, and we'll be done in no time. So
okay,
I'll do as many of these as you want, because I enjoy talking to you. I don't see any animosity in it. I enjoy it, understanding each other's position. So, yeah, why don't we call it a day today and reconvene at some point?
We got the iPad here, and we can talk about alleles, basically, and loci, that sort of thing at some point, and we can just draw. You can see my screen, right? Where I'm drawing.
Oh yeah, that's some good artwork. My daughter was looking at that, going, "Wow."
I am so good at this artwork.
I'm impressed more and more every single day.
Kind of a neat program, actually. It's
that's cool.
My company makes this. It's called Linea.
Oh, really?
Yeah, and it's used by a bunch of high-powered people in Hollywood, actually, to do some artwork.
Nice.
People do really good art on this thing because you get all sorts of brushes.
I may have to invest in that program. Not gonna lie.
I think it's only $10 or something. And
that's not bad.
And I think I get like 30 cents if you buy one. So
all percent. You've earned it.
We will get into that, and we'll talk about loci and stuff like that. But yeah, I mean, we can, and then the overview stuff. The stuff to take away from this is that you and I have a very, very different scientific background, and we have a different set of facts underneath us.
Right.
I have found that I think you're very confused about this heterozygosity. But anyway, don't take that bait. Don't take that bait.
Trust me. Because I gotta go to the washroom from these 3 bottles of water, I ain't taking no more bait.
Yeah. Can you please send me anything you got on Carter? And
yes. I do want you to look it over, because it's beyond the scope to go like he goes in, and I think you're going to enjoy this because it's pretty much exactly what he does, a full computer modeling of biblical genetics, starting with the created heterozygosity. He goes over the amount of alleles that would be present, and then he goes over the current genetic data when it comes to SNVs in the world population, the insulin one. But yeah, I don't want to go any more rabbit trails. I'll send you those. You send me anything you want. This was a good conversation. I enjoyed it.
Me and I'll put them in the links, and that'll be cool. There's something else I was gonna say, but I can't. I lost it. Anyway, but yeah, we want to put everything in my descriptions, that we talked about, and I'll put some elaborations. I'll try to put some in Jensen's papers, and I will look at. Oh, what I wanted to say to you is that if I find out that you're right about any of this and I'm wrong, when I look into it, I am going to become your greatest ally. I know.
That's why I'm working so hard to convert you.
Yeah, but that ain't going to happen.
Hey, you never know. What are the as Dumb and Dumber, Jim Carrey said, what are the chances of a guy like you converting to biblical creation?
Yeah.
What are the chances? 1 in a million? 1 in a million.
Yeah. I'm guessing 1 in about 100, but we'll see. You got a long way to go, man.
You're telling me there's a chance.
What's that?
So you're telling me there's a chance.
Oh yeah, okay. There you go.
Hey, and if you convert me, I promise I'll be a really good ally to you as well.
I think you're going to be amazing when you cross over to our side. Absolutely amazing. Okay. Well, thank you for doing this. It's been very pleasant. I enjoy you. I tell everybody that mentions you or criticizes you that you're actually a pretty great guy. So
awesome. I appreciate that too. I got kind words to say about you. Like I said, I enjoy our conversations. I find you funny every time I listen to your broadcast. I always get a kick out of your assessments, and you know that because I comment typically. So yeah,
okay,
keep it up, man.
Because you know them, Raw Matt and John Maddox. Tell them it's not personal, but
I will. We'll get them on your show too. And in the meantime, be there for 8:15. Should be a good discussion tonight with Ron and John. So I'm looking forward to it just in a couple hours. So
right. Okay.
Okay, man.
We're going offline.